Hi everyone, welcome to 𝗖𝗵𝗵 and today we are discussing general microbiology. In general microbiology, we are discussing the specialized structure and this is the 8th class. So in the anatomy of bacterial structures, the very first structure which we are going to discuss is flagella. Flagella, as you all know, is the organ of locomotion and it represents H antigen. In my class on cell wall, I have told you EFGH the mnemonic and F and H go together. That means flagella is associated with H antigen. Now flagella is made up of what? It is made up of flagellin protein. What are the parts of flagella? So you have got a tail, you have got a hook, you have got a basal body. These are the three parts of flagella. And the basal body, for that matter, in gram positive, it is made up of S membrane, M membrane. Whereas in gram negative, it is made up of lipopolysaccharide, LPS. I have told you, gram negative, LPS layer, common relation, S membrane, M membrane present in both of them. It's just the peptidoglycan which is there in gram negative bacteria. Okay. So the mnemonic is sample SMPL, that is S membrane, M membrane, P and L, that is present in the gram negative. Now what is the different arrangement of flagella? So flagella can be monotrichous, that means it is present only on the one side. So I am writing Vibrio like this so that you can remember that it has got flagella only on the one side. So Vibrio has got flagella on one side. Next is Lophotrichous. Lophotrichous means that it is present on the one side like this but in a bundle. So Lophotrichous. Have you seen helicopters? Helicopters have got their wings only on the one side. So Helicobacter, just like helicopter, is having the flagellar arrangement on one side. Amphibians. Have you heard this word amphibian? Amphibian like frog. So amphibian like frog means which are organisms, those which can live in two environments, the land and the water. So amphi means both. So Amphitrichous means they have got flagella on both the sides and spiral in shape. This is spirulum. Peritrichous, that means present on all the sides like this. Peritrichous everywhere around. So peri, P and R for Proteus, E for E. coli and peritrichous last S for 𝗖𝗵𝗵𝗵, Salmonella. Salmonella, you can remember always Salmonella is 𝗖𝗵𝗵𝗵 and 𝗖𝗵𝗵𝗵 has got public all around. So that's why it is a peritrichous. Coming on to atrichous, that is non-motile. So who is the one who never dances? 𝗲𝗵. So 𝗲𝗵 will not move. Okay. And Staphylococcus, only eating grapes, love for grapes, not dancing. Okay. And if you don't know what I mean by eating grapes, somebody in the comment section please comment and let other people know what the story of Staphylococcus and grapes is. Okay. Coming on to the motilities which are based on flagella. So you have got darting motility. So darting motility is shown by Campylobacter jejuni and Vibrio cholera. So basically there is different style of walking. Okay, there is different style of walking. Like we boys, we walk like this. Girls walk like, okay. Similarly, bacteria has different styles of walking. So darting motility is one of them. So the mnemonic is C and D coming together. So darting motility, C for Campylobacter and cholera, they show darting motility. Tumbling. So have you seen the tumblers which are being used by small children? They have got a list, right? They have got a list of 100 ml, 200 ml and everything on that. So my dear friends, list on the tumbler, that means tumbling motility by Listeria. Coming on to corkscrew. So have you seen corkscrew? Yeah. So something which can screw the life of bacteria is TLC, that is total leukocyte count, can screw the life of bacteria. WBCs can screw. So screw. So corkscrew has the mnemonic TLC, L for leptospirosis, T for treponema, C for corkscrew. Lashing. Lashing is by the 𝗵𝗺𝗺, Borrelia 𝗵𝗺𝗺. So dekho, lashing means lush. Lush is shining. And which is the shining car? Ferrari. So Ferrari, Ferry. Ferrari, Ferry. That means lashing Borrelia 𝗵𝗺𝗺. So lashing, that means shining, which is the shining car, Ferrari car, that's it. Swarming is by the Proteus. So Proteus, the end word S for swarming. 𝗓𝗵𝗄𝗏, 𝗓𝗓𝗏𝗏. So Clostridium tetani. Okay. So you can remember PQRST, SNT coming together, S for 𝗓𝗓𝗏𝗏, T for tetani. And in order to demonstrate, I've already told you in the stains which are starting with L or having L, Leifson's and then silver impergenation, because flagella has got a lot of L's in it. Cool. Coming on to Fimbriae. Flagella we are done. Now we will discuss about the Fimbriae. So Fimbriae, I have already told you it is an organ of attachment. If you remember, I told you it is responsible for causing CAUTI, catheter associated UTI, because it is an organ of attachment. This is a previous year question also. Fimbriae is going to show heme agglutination with human RBC. So it can lead to agglutination of RBC, clumping of RBC. Okay. A specialized Fimbriae, it is called as Pili, and I have told you in bacterial genetics video that it has got a role in conjugation. Pili or Fimbriae, these are seen under electron microscopy because they are very small in size, 10 to the power minus 9 meters or 1 nanometer, something like that. But you don't need to worry, it's not important. Coming on to the next part, Glycocalyx. In the very first video, I gave you a little introduction to what Glycocalyx is. So please understand, Glycocalyx is for the adherence and it has got an anti-phagocytic property also. If you remember, I told you that Glycocalyx or capsule, you remember capsule I have told you? So Glycocalyx, if it is well organized, then it is called as capsule. But Glycocalyx, if it is loosely arranged, it is called as slime layer. But both of them have a function of adherence and both of them have a function of anti-phagocytosis. And I have told you a very beautiful song, AVSM and capsule, right? So whenever you have capsule, AVSM should be on your fingertips. A for anti-phagocytosis and the other one is adherence. And I have told you that every organism is going to have polysaccharide except polypeptide, that is by the glutamic acid and Bacillus anthracis. I have told you polypeptide, P, or even leave this mnemonic, just remember I have told you the mnemonic bag, that means glutamic acid, and glutamic acid is a protein, that is why we say it is a polypeptide, whereas all others have polysaccharide. Don't miss out the cute 2 C's of polysaccharide because this will remind you about the coccus. So what are the example of coccus? Meningococcus, Cryptococcus, Pneumococcus, okay, all the coccus. And mind you, Streptococcus or the Pneumococcus and Haemophilus influenza, they both are real brothers. Wherever Pneumococcus will come, H. influenzae comes almost all the times, okay. And then you have got 2 bacteria, non-motile bacteria. Do you remember? Klebsiella, Salmonella, Salmonella and Klebsiella. Okay, you got it? Great. Coming on to, just a second, Klebsiella. Yeah, Klebsiella is non-motile, not the Salmonella, right? Salmonella is not, Salmonella is motile. So you can just remember you have got Klebsiella, and from the S of the polysaccharide, you can remember the Salmonella. Last but not the least, you have got hyaluronic acid and Streptococcus pyogenes is there. Okay. Coming on to the functions, I always teach you AVSM for capsule. So AVSM means A for antiphagocytic, V for virulence. That means it is used for the purpose of manufacturing of vaccine. Serotype production, different serotypes I have told you. And M is mucoid colony formation. This I asked in one of my previous videos. And yes, you all replied and thank you so much for that. Okay. So pick the mnemonics, guys. Pick mnemonics, Spurs mnemonics, very important, right? So this was all for the capsule. Coming on to now cell membrane. Just leave this numbering. Coming on to the cell membrane. Like any other cell membrane, it is a semi-permeable membrane. It contains enzyme permease, which permits the transport. It allows the influx of nutrients and efflux of toxic metabolites. It also has a role in cell wall synthesis. I told you membrane is just like mom, like mother, right? Coming on to mesosomes. So mesosomes are the extension of cell membrane only. Mesosomes are the extension of cell membrane only. And the septal one, the middle one, is required for fission. So can you see, if it is in the middle, it can make a cut and there will be two bacteria formed out of one. And if it is lateral, it is for respiration. If it is lateral, it is for respiration. Coming on to the storage granules, I have already told you that diphtheria is the one which is responsible for, storage granules are formed by diphtheria. Which will be the stain which will be used? That is your question, because I have already discussed in a previous video. These storage granules are also called as volutin granules and metachromatic granules. Why metachromatic? Because these are differently colored. Just like your food is differently colored, these are also differently colored storage granules. So chromatic means color and meta means different, so different color. And volutin, you can remember that these storage granules are just like your big tin boxes used for storage, so volutin. Okay, like that you can remember. Coming on to this beautiful diagram, and yeah, this is important. So see here, these small ones, plasmid, we have already discussed in genetic video. Then next you have the nucleoid, this center one, then the ribosome, these ones, cytoplasm, this amorphous one, Fimbriae, the long one pilus, and the longest one is flagellum, then outermost capsule, then cell wall, and then the plasma membrane. Coming on to bacterial spores. So my dear friends, spore has got various layers. Okay, I'll teach you with the help of this analogy of a room or of a home. So you have got a room that is called as core, okay, that is similar to core. Then after it, you have got a balcony, like one room, second room. In middle of both of them, you have got a balcony. This is called as cortex. Okay, so this is called as what? This is called as cortex. After that, you have got a wall. Okay, so 4 walls of your house. Okay, so that is a wall. So you can remember that corresponds to spore wall. After that, you have got a coating, the paint coating on the walls. So this is called as spore coat in the bacterial spores. And in the end, you have got external surroundings, exosporium. So what I told you, the room is like a core, balcony is a cortex, then you have 4 walls of your house, then the paint coating and the exit exosporium. This exosporium is important to provide resistance to the spores. That's why even in the adverse conditions, the spores can survive for longer duration. And the stains used are Muir stain and Fulton stain. You can remember this. Okay, so Muir stain and Fulton stain. So Muir, yesterday I told you for the capsule, so same are there, capsule and spore. Fulton you have to remember. So just like the spore, protect yourself from the adverse conditions and keep on growing and have a beautiful survival on this beautiful planet. Thank you so much. And yes, the comment section is open for your feedbacks, for your love, blessings, care and support. God bless you. Thank you.